We are all coming from different backgrounds and we work in different places of the world.
I'm developing a new drug which will improve the diagnosis and treatment of cancer.
Treat people and not give them side effects.
MEDICIS-PROMED has a big strength, that is the diversity.
The advancement of medicine is closely linked with that of chemical science, and in particular the study of the potential of all the chemical elements and their isotopes in the periodic table.
Isotopes are very important in medicine in general and have been used since 80 years, because we can label these isotopes to any molecule virtually that we inject into the patients and we track into the body of the patients. All this comes from the discovery of radioactivity made by Pierre and Marie Curie, which led to the discovery that isotopes could be made artificially radioactive. Over the years, we have expanded our tools in terms of machines, technology, and biological understanding of the diseases.
Although known for almost a century, this area of research is still in its infancy. Future advances in the design of new drugs and diagnostic agents require more knowledge, new methods, in essence, more research.
The more isotopes we have, the more molecules we have, the more radiolabel probes we can produce, the better we can meet the specific needs of the patients in terms of diagnosis and therapy, which, in other words, we call personalized medicine.
MEDICIS-PROMED is a European Marie Curie training network. It involves private companies, research institutions and universities. We are 15 PhD students, all coming from different backgrounds. We are medical doctors, engineers, physicists, chemists.
A network of young PhD researchers from such diverse backgrounds is the smart solution to tackle the many challenges involved in developing new radiopharmaceuticals from the untapped potential that the periodic table of elements contains.
I work on uranium carbide target material. It is one of the best target material known to us, but for medical application, we need a better production yield of radioisotopes, where conventional target material doesn't fit this requirement. So here I am working on uranium carbide target material at nanometer scale for better production of innovative medical radioisotopes for clinical research.
To develop a target, I'm developing a new production method for the production of non-conventional isotopes. The aim is to make available isotopes for medicine that are right now not produced or produced in not sufficient quantities. Here at CERN, we are preparing the targets that are irradiated at MEDICIS. So here is arriving the proton beam, hitting the target material inside, where radionuclides are produced.
So in our project, we are trying to produce the medicament which will correspond to this highly precise medicine. So with my jobs, I need to highlight with lasers the desired isotopes, which will be after extracted. Using our separator, we are producing the radioisotopes, which is highly purified from any kind of contamination. And in this case, we will be sure that our radioisotopes, with these properties which it has, will target only the cancer tumor and will not damage the living tissue. So we can treat people and not give them side effects.
We are producing these isotopes here and we collect them, and then the idea is to purify these isotopes. So we prepare this for shipping to hospitals or research centers. And of course, we need to ensure the quality control of the product.
The goal of the chemical process is to purify the medical isotope from the impurities it comes with. The sample is first dissolved in acids, and the resulting solution is passed through a column that contains a specific resin. The resin captures the medical isotope, while the impurities simply flow through. Then, the medical isotope can be retrieved from the resin by changing the chemical environment, as isotopes don't stick to the resin anymore. The purified isotope is collected in a vial and shipped to hospitals for radio labeling.
The challenge of this project is that once collected and then chemically treated, the radionuclides can assume different chemical forms. They can be solid or liquid, and then they can also vary in terms of quantity. So that kind of container should allow the transport of solid and liquid sources, and also big quantity of them.
In the chain of isotope production, I'm an end user. My work starts from the patient. My experiments help to define the properties of isotope and where exactly it could be used.
I synthesize new molecule and I label with the radioisotope at research level, and then we can test these molecules preclinically in cells or in animals and see if they can be useful for a future clinical application.
I take the radioactive isotopes that comes from CERN, I label it to a molecule, and I do all the preclinical testing to prove that it works and that it's safe.
I use the isotopes produced at CERN, incorporate them into a specific navigation system, and which will be used to diagnose or treat cancer. My particular probe will improve the early stage diagnosis, which will also improve the survival rate of the population. Because we use it only for mice. For mice we don't.